Hello and welcome to Healthcare Voices, where we bring you authentic voices behind healthcare innovation and impact. I'm your host, Catherine Cummings, former clinician turned healthcare technology advocate. And today I have the privilege of speaking with Professor Travis Nemcoff of CU Anschutz Medical Campus. Professor Nemcoff is an accomplished researcher in metabolomics and cellular metabolism, making groundbreaking insights into how these processes impact health and resilience. His work, especially on red blood cell metabolism and elite athlete physiology, explores the role of metabolomics in precision medicine and chronic disease management. I'm so excited to learn about how his research can transform healthcare. Thank you so much for joining us and welcome to the podcast.
Thanks for having me.
I'd like to learn a little bit about how you entered the field of metabolomics. What really initially inspired your focus on understanding the intricate world of cellular metabolism?
Yeah, so I guess to go way before entering metabolomics and broadly metabolism research, I did my undergraduate up at CU Boulder, and I was a cellular biology and biochemistry majors. And I'd gotten a job at Roche, had a manufacturing facility up in Boulder at the time, making peptides, which are sequence of amino acids that have bioactivity, and our body makes them, and lots of drugs are peptides. Maybe one of the better known ones is insulin, is a peptide. So I got this job building peptides. It's all chemistry done in these reactors, and that led me, after that internship over the summer was over, to a neuroscience lab at MCDB in Michael Stoll's laboratory, and he studied Alzheimer's disease and how that disease really sets in, the manifestation of that disease. And to do a lot of the studies he needed to do, he needed to make a large quantity of a specific peptide called beta amyloid that was very difficult to make. So because of my background, I started coming up with a scheme to make that peptide with Michael's assistance. And we ultimately came up with the scheme to make a peptide recombinantly, which means using engineering bacteria to grow it for us. And then there's a variety of different purification schemes that you can do to clean it up and get highly pure amounts. And it turned out that process worked to make a lot of other peptides, including insulin and a variety of modified insulins and a whole host of different therapeutic drugs. So we had spun that out of his lab to start a company called Amide Bio out in Boulder, Colorado. It's still around today, and I worked there for a few years. But I started thinking about the larger scope of biology, the systems-wide scale of biology. Up until that point in my career, I was really just focused on this very narrow application of making this singular reagent, which gave a good skill set that would have been translatable into to pharmaceutical manufacturing, for example. But I had a bachelor's degree. I wanted to start guiding my own research. So I wanted to go back to graduate school and get a PhD. And now with this new understanding of what I really wanted to do, looking at biology from a systems-wide holistic level, became interested in omics technologies. And the that suffix basically means the system. So the genome is the system of genes. And the proteome, which is what I had become interested in at the time, is a system of proteins. And so there was a lab down at CU Anschutz run by Kirk Hansen that was doing a lot of interesting work looking at using the technologies in the field of proteomics to study the extracellular matrix and how that composition and the biomechanics of the extracellular matrix can influence tumor metastasis and tumor growth in general. So I joined that lab and started learning proteomics and mass spectrometry, which is the technique that we use specifically in the lab to measure all the proteins in the system. And Kirk at the time was thinking about venturing off into this newer field of metabolomics. And so I was trying to decide, well, I want to do something systems-wide and holistic, so proteomics works well, but metabolomics had a lot more low-hanging fruit because it was a very young field at the time, and there wasn't a lot out there. Before we got on the podcast, I looked it up. So in 2012, if you go on PubMed and look for the number of publications with proteomics in the name, there's about 9,100 publications, and there were 2,400 publications in metabolomics. So I thought it was an opportunity to get into a field at an even earlier stage. Throughout my process of working with lots of peptides, learned that working with small molecules, which are metabolites, they're easier chemically to handle. So I thought, I'm going to go into metabolomics. And around that time, I also read a quote from James Watson, who was one of the scientists, along with Rosalind Franklin and Crick, to solve the structure of DNA. So he's won the Nobel Prize, very well-known scientist. And he had said, about around 2010, if he were doing his PhD today, he would have done it in metabolomics. So then I was sold, and that's how I ended up in metabolomics.
Well, that's a really cool story, and it really sounds like you've worked in a lot of different fields. I'd love for you to give us an overview of some of your current research areas, particularly in relation to metabolic health and cellular metabolism.
Yeah, so my PhD thesis was in red blood cell biology, and we really love red blood cells down here. It's a perfect cellular model to study metabolism as red blood cells mature. So they start out in the bone marrow from a stem cell, and then they go through a series of maturation steps. And that process is very unique in the red blood cell because it gets rid of a lot of the compartments in the cell, the organelles that other cells would rely on. So specifically, I'm talking about the nucleus and the mitochondria. They get rid of all of those because they need to maximize the amount of space within this very small cell to have hemoglobin so that they can circulate and deliver oxygen throughout the body. So what that really means is that metabolism is the primary mechanism of cellular control in the red blood cell. And so it's a great model to study metabolism in general. And a lot of the concepts that we've learned through studying the red cell are now being observed in cancer cells, which have a nucleus and other organelles. So that's, kind of how we had gotten into it. The other interesting thing about the red cell is of the 30 trillion cells, estimated 30 trillion cells in our bodies, 25 trillion of those are red blood cells. So it's by far the most abundant cell in the body, and it travels throughout the whole body, and it sees most of the body. Anywhere there's blood, there's red cells. So it interfaces a lot of the system's homeostasis in addition to just delivering oxygen. There are studies showing how it interplays with the immune system and how the red cells interplay with controlling vascular tone, how dilated or constricted a blood vessel is. It influences blood clotting. So it has a large number of roles in addition to oxygen delivery. And that is, so that's really where our research lies. And we study red cells. I work with Angelo D'Alessandro, who's become a world leader in red cell metabolism. A lot of what we are studying is how red cells behave in health and in disease. And so from a healthy standpoint, we're talking about high altitude acclimatization, exercise adaptations, and then from disease, anything from various kinds of anemia, like sickle cell anemia, to aging and various cancers. The red cells can be impacted throughout those entire realms. And so that's really largely where our research lies. And then medically speaking, we can talk about more of this as we get into it, but transfusion medicine is a huge need for red cells. It's the second most used medical procedure, I think, behind vaccination is red cell transfusion. There's 10 million of red cells transfusions a year. So it's a very commonly applied medical technique, and red cell blood banking. There's a lot that is we are learning about how red cells behave in the blood bank and how you can intervene to optimize their quality of storage and efficacy during a transfusion. And so that's a large part of our work too.
That's a great lead into my next question. Are there any particular discoveries that you've made about red blood cells that could influence how we understand or treat certain diseases? You've mentioned a couple.
So yeah, it's a good question. So I think the first one is that comes to mind is altitude acclimatization. So it's been known. I remember in my biochemistry class learning about allostery, which is a mechanism in biochemistry where a small molecule will stick to a protein and influence an enzyme or a protein and influence the activity of that enzyme, and because it changes the shape of it and influences how that enzyme works. And the example that is used when they teach allostery is diphosphoglycerate or bisphosphoglycerate. It's the same molecule, two names. So bisphosphoglycerate is a molecule that binds to hemoglobin, and it stabilizes the deoxygenated state of hemoglobin. It promotes the ability of hemoglobin to release oxygen. So that's very useful in hypoxic circumstances. What was found in, I believe it was maybe the 40s, 50s, and 60s when these papers were published, is that one of the immediate adaptations to high altitude is that the red cells start to increase their production of bisphosphoglycerate. And so that was the original mechanism that was commonly taught to teach allostery in biochemistry classes. But when we got involved, there was a study going on campus called altitude omics, which was a similar suffix, the omics of altitude acclimatization. And so they had collected various kinds of omics data, genomics and transcriptomics and proteomics and lipidomics and metabolomics on individuals who had never been to high altitude before. They collected all these data along with physiological tests and memory, how long it takes to run a 5K, sorts of parameters were determined with these individuals. And then they took them, I think this was in Eugene, Oregon, at sea level, and then they took them down to Bolivia and they drove them up to the top of Mount Chacaltaya, which I think is 17,000 feet or so. And they lived up there for about 2 weeks. And so samples had been taken throughout this whole process, and then they came back down to lower elevation, and then they went back up to high altitude again 2 to 3 weeks later to understand the molecular mechanisms of memory, acclimatization memory, which can be easier to acclimatize the second time you go up to high altitude. So we got involved in these studies after they had been initiated, and we were responsible for running the lipidomics and the metabolomics. And through interactions with colleagues at University of Texas, Yangsha's lab, she's in China now, but we actually understood the mechanism of bisphosphoglycerate accumulation. So it had just been observed that it happens, but there's a signaling network that elicits the production of this particular metabolite in the red blood cell. And that signaling network is actually involved in pathogenesis in sickle cell anemia. So in a sickle cell, individuals who have sickle cell disease, when the hemoglobin delivers an oxygen molecule and becomes deoxygenated, there's an amino acid substitution that changes the shape of the hemoglobin, and it causes it to sickle, causes it to polymerize, and the cells ultimately will sickle. And so that's where the name comes from. When you look at a blood smear, the red cells are in a sickle shape. And it's because of the single amino acid mutation. But that network of acclimatization, that molecular signaling network that tells the red cells that there's less oxygen around and they need to change their behavior, can actually contribute to the pathology of sickle cell anemia. So by interfering with that network through a variety of different techniques, that's one angle now that's being looked at to treat sickle cell disease, by scavenging some of the molecules that are produced through that circuit. So that would be, I think, an example of how what we're studying can impact treatment. I think that was your question. That was a long answer.
Yeah, that's really cool. I mean, it seems like there's the opportunity to make a really big difference for some of these patients, and sickle cell anemia is one of those that you learn in medical school and nursing school for all of the clinicians, that it can just really impact quality of life, especially for young children, pretty significantly.
I do have one. Yeah, I'm sorry to interrupt you, but I do have one other answer that you might want to.
Yeah, I'd love to hear it. Let's hear it.
So another area that we're working on, like I mentioned, is transfusion, and transfusions are very common, right? You have millions of Americans alone in the country are donating blood altruistically to feed this blood supply, of which there's becoming a shortage. And there's a high need for transfusions for all sorts of diseases, including sickle cell anemia, or other diseases where you need a chronic transfusion to replace red blood cells, or in acute instances like a car accident where somebody's lost a lot of blood and needs that blood replaced. But this is a biologic treatment. I mean, we rely on human donors for these blood cells, and not all humans are the same. I don't think that's a controversial thing to say or a surprise, that our genetics and our behaviors and our exposures can influence how our bodies function. And so to understand better the role that human donor biology plays in the impact of or the quality of the red cell, that is how well it stores and how well it transfuses. The NIH had set up a study a while back called the Recipient Epidemiology Donor Study, or REDS. And that study was a massive study that collected blood donations from 14,000 people, and they stored them. Those were transfused. About 6,000 patients received only that one blood unit, so they were able to monitor how well that blood unit did in a patient. The genetics of those donors were determined, and we looked at the metabolomics as well. And so now what we're starting to find, and which is really interesting and cool, is we're able to stratify specific metabolic signatures as a function of a donor's age or sex or BMI or genetic ancestry, or various exposures like caffeine, or if they're smokers or non-smokers. And from this information, we're now coming up with new possible ways that we can improve storage quality. We can see that donors with certain characteristics lack a specific nutrient. We may be able to add that nutrient into the blood bag, into the liquid formulation that helps sustain that blood cell as it's stored in the blood bank over time. So I think these studies are great because, one, we're not advocating that any blood donors should be deferred. That's not needed. I think blood transfusion largely is a success. It works very well. And what we're trying to do now with our studies and our platform is try to come up with ways where we can make it even better.
Very cool. I think the next thing I'd like to know, there's a lot of new tools, techniques, it sounds like, that are coming out. Are there any specific tools or techniques that you guys have enacted in the lab that have really transformed how you work or how you receive data from?
Yeah, I think from two specific angles, from acquiring the data and then analyzing the data. So recent innovations in both of those areas have had a massive impact. So from the data acquisition side, like I said, we use mass spectrometers. These are really fancy, expensive instruments that sit on top of a bench and they will weigh. They're essentially really expensive balances, and then they can weigh the molecules provide weights of molecules that are in complex mixtures, and they also provide the amounts of those molecules in complex mixtures. And there's a lot of physics that goes into taking those measurements, but to distill it down simply, that's essentially what they do. So you can measure all of the metabolites in a specific sample and how much of each of those metabolites resides in a specific sample. The instruments have gotten a lot more sensitive. They've gotten a lot more accurate and a lot faster. And the speed in particular is really important for us because it allows us to run really large studies where we can now start covering a reasonable degree of a population, either a donor population or a patient population. And of course, there's always power in numbers in medical studies. The more patients and donors you can look at, the more variability you can cover and characterize and quantify and understand. So these instruments, the fact that we can start running them faster has really taken us to another level and allowing us to power our cohorts and our studies much larger so that we can gain more insight. But with that comes much more data, complex data, and more of it. And so artificial intelligence and machine learning has really started to help us quite a bit because when you apply various algorithms, machine learning algorithms to these large data sets, we start to recognize patterns that we didn't necessarily see were there. We had a feeling that they were there, but now we have a better definition and a characterization of those patterns. And so this newer era of AI has been a big advantage for the types of studies that we're doing too.
I was curious about whether or not you guys utilized AI. We're hearing a lot about that, especially with vaccinations. They're able to analyze large data sets, and the speed to getting, the new flu vaccine strain has been faster, things like that. So it's cool that you're using that to analyze your data as well. A couple of things that you mentioned that I think that the audience would love to hear is, you mentioned tumors, you mentioned kind of tailoring treatment a little bit. How might your work contribute to, I would say, like the growing field of precision medicine, particularly for tailoring treatments to individual metabolic profiles that you mentioned?
Yeah, it's a good question. It's something we've been thinking a lot, quite a bit about recently. I think what we do is going to be essentially very useful is twofold. One, using our techniques, we can better characterize mechanisms of action of drugs. So we can better understand how drugs are working, specifically and especially, I should say, from the metabolic level. So having a better characterization of how drugs work will hopefully improve medical practice. The other way that we're hoping metabolomics can contribute to the precision medicine goals and initiatives that are widely held, I think, across the industry, is through stratifying patients in a much more precise way. And so the term that's used a lot is endotyping, identifying subgroups of patients based on molecular profiles that can potentially determine their disease trajectory, better qualify or quantify their likelihood of having certain comorbidities eventually show up, certain kinds of symptoms eventually show up. And when you talk about, like diabetes, I think there's like 70 different kinds of drugs that can be prescribed for diabetes. And then you've got hypertension. There's tens or 30 drugs for hypertension. And how do you decide which combination of drugs and at what timing will work better? And if you can start to acquire more data on these patients, and specifically as it pertains to their metabolism, we're hoping that we can identify subgroups that would have responded to a specific treatment regimen more readily or quickly than another regimen, or another subgroup, which would get a different type of treatment regimen. So that's basically from improving our knowledge of how drugs work on one side, and then improving our knowledge of which drugs are best for which patients on the other side, is how we're hoping to improve healthcare.
Before the podcast, you shared with me that you're doing some research on athletes, specifically elite athletes. I'd like to understand more about what kind of unique metabolic markers or adaptations that you have observed in these high-performing individuals.
Yeah, it's been a cool series of studies. We have spent a lot of time working on disease. I think it's just taken for granted what health is. So we started to understand or wonder what is health from through the lens of how we are taking measurements. And so to do that, I got in touch with a colleague who is a performance coach, Inigo San Milan. And he works with a lot of elite athletes. And so we were able to run some of these studies with elite athletes. Started out with cyclists, and other studies I've now been working with ultra marathoners and ultra runners as well, which is a different type of athletic paradigm. But these are all, predominantly elite athletes. And so what we've learned first and foremost, which isn't necessarily a surprise, was the degree to which they're metabolically flexible. So this concept relates essentially to the types of fuel that the body can use to generate energy during exercise. And elite athletes in particular, have this supreme capability to keep burning fat at higher levels of intensity and for longer periods of time than the rest of us. And that switchover point between burning fat and burning carbohydrates really comes down to the demand for ATP, adenosine triphosphate, which is a molecule that carries energy throughout the body. So the demand for ATP versus the rate at which the body can supply that ATP. And when that demand exceeds the supply, the body will switch over to burning carbohydrate, which generates ATP a lot more quickly. But you start to fatigue more when you switch over to that other process. So elite athletes can maintain their ability to not need to switch over and tap the carbohydrate reserves for a lot longer and that higher exercise intensity. So that was one major thing that we've characterized. And I want to say characterized rather than learned, because it was well known that can happen, but you needed to measure these types of things with like a metabolic cart with respirometry in a centralized lab facility. They put a mask on and ride on a bike, and then they measure the amount of oxygen consumed and the amount of CO2 produced, and you can infer a lot of this information. And what we did is translate that technique to just doing it from a single blood drop that you can take from a finger prick, or we have these devices you can it's like sticks to your arm and you push a button and it will take blood drops out, and we can measure it that way. So that was the first thing I think that we understood studying elite athletes, is that we can measure it in much more diverse contexts. So now we can follow them through races, and we've done that. We've taken blood samples throughout multi-stage races, and then we can bring them back to the lab. And we found that the cyclists that performed better were maintaining that fat burning capacity for longer throughout the race than their peers who had switched over to carbohydrate burning earlier on in the race. And so that was, I think, maybe the first time that had been measured in the field in real time. We have another, number of other metabolic adaptations. Even when you look at these elite athletes and you try to separate them based on their metabolic fitness, which we measure as a lactate threshold, the athletes with less metabolic fitness, even though they're world-class athletes, just through that lens with less metabolic fitness compared to the ones with better metabolic fitness, have differences in their metabolism. The better athletes appear to synthesize coenzyme A more readily. This is an important molecule that traffics atoms throughout cells and into the mitochondria and out of the mitochondria throughout the cytosol. And it's built from vitamin B5 pantothenate or pantothenic acid. So the better athletes seem to have a more active pantothenic acid catabolism or coenzyme A synthesis pathway. They also have a different signature in branched chain amino acid utilization. There's a lower level of branched chain amino acids in circulation after exercise in the better athletes than the not as good athletes. I don't want to say worse athletes because they're still world class. And there's also appears to be a difference in oxidation profiles. The better cyclists actually have higher markers of oxidation in their blood, which is a little bit counterintuitive because, oftentimes you hear, well, if you exercise, you're supposed to take antioxidants because oxidation is bad. But like many things, oxidation in biology is a spectrum, and there is a certain degree of oxidation that's a good thing. And it can signal to the rest of the body to grow and repair itself so that it's even stronger than before the exercise began. This is part of how the body adapts to exercise. And so that was an early observation, and we can't speak too much to exactly where the oxidation is occurring, to what extent the oxidation is occurring, but we did see markers of higher oxidation in the better performing athletes, which might mean that they've got a better regenerative capacity or recovery capacity or antioxidant capacity that helps them recover within their own bodies. But that's for future studies.
Very interesting. I mean, it seems like the elite athletes just generally demonstrate a unique physiological resilience that you or I might not exhibit, right?
I mean, I have done the same metabolomics test on myself that we've done with elite athletes, and I can graphically and quantitatively now say just how much better they are than me. It's pretty.
Well, along those lines, are there any specific metabolic adaptations that you've identified in studying elite athletes that maybe could inform treatments for the non-elite, right? Or, for those that have metabolic conditions or are suffering from things like oxidative stress?
Yeah. So for as far as metabolic stress, so one population of patients that we work with are cancer survivors. So this is in collaboration with a program at CU called Be Fit, Be Well. Dr. Ryan Marker runs this program. And so how it works is patients who are treated at the cancer center, CU, University of Colorado Cancer Center, are eligible to join the Be Fit, Be Well program after treatment. And it's a 3-month exercise program with a personal trainer about 3 times a week. And exercise oncology is really interesting field that is growing quite a bit now, because after a cancer diagnosis, exercise is one of the best things that can be done to maintain quality of life if a person is able to. Maintaining activity and exercising is very important. There's recent emerging evidence that also suggests it might actually impact how well treatments could work in certain kinds of cancer. It might influence the chance of relapse and decreasing the chance of relapse. These are early studies, and it's not going to be for every type of cancer and for every type of patient background. But it's very interesting and very exciting. So we started working with this program and implementing some of the same platforms that we developed working with elite athletes to try to monitor exercise, the impact of exercise on metabolism in cancer patients. And so it looks preliminarily like in the first set of patients that we worked with, that the amount of fat circulating in their blood at baseline is lower after they finish the program. And that would suggest that that exercise program improved their body's ability to burn that fat, improved their metabolic flexibility. So started moving them towards this adaptation that we talk about with elite athletes. Not all patients show that adaptation. Not all participants in the program showed that. So if now this is an example of personalization of therapy, we can monitor some of these more precise markers to see if that exercise prescription should change. Should they be doing a different kind of exercise for a different period of time, so that you can start moving them towards what we know to be a more healthy version of metabolism.
That's exciting, though. I mean, I think everyone's been impacted by cancer in some way, right? And everyone's always looking at, what they can do to prevent or, prevent relapse, right, of cancer. The last question I have for you is, where do you see the field of metabolomics heading in the next 5, 10, 15 years? And what are some potential breakthroughs that you're most hopeful for?
I'm really excited and hopeful that metabolomics starts playing a more integrated role in clinical chemistry. So right now we go to the doctor and we get our blood drawn and sent for metabolic panels. But these metabolic panels don't contain a lot of information about the metabolism that we measure. They'll contain inferred information, amount of transaminases that are circulating in the blood, or creatinine, or sodium and potassium. But what I'm excited for, because we have the capability in small amounts of sample, we're talking a few blood drops, of measuring hundreds to thousands of metabolites. And if we can now start applying these sorts of technologies and integrating them into standard clinical chemistry workflows, I think there will be a lot more information for doctors to treat their patients better, a lot more information for patients to understand their own health better. And so I'm really excited about that, but it takes time, because you want to be very confident in the results that you're reporting to people. So this is going to require a lot of QC and QA, standardization of how samples are collected and handled with medical professionals and with the biobanks that are collecting the samples for studies themselves. And then there's regulatory approval that's required to move these forward. So there are a few companies out there now that are using multi-omics, metabolomics, proteomics, genomics, lipidomics to build these next generation tests. And it's early days and it takes time to really validate them, but there's some successes are starting to emerge, and I'm hopeful that in the next 5 to 10 years, we'll see an integration of these technologies into standard healthcare practice, because I truly think that it's going to improve not only how we treat healthcare or treat disease, but also preventing the onset of disease and compressing that morbidity curve that impacts health span.
Thank you for sharing all of that with us. I appreciate the time, and it was a pleasure speaking with you today, and I really appreciate you joining me on the podcast.
Yeah, thank you. It's been great. I really appreciate the chance to share and the invite.
Thank you to all of our listeners for joining us on the podcast today. We appreciate your support. To hear from more healthcare voices like Professor Travis Nemkov, hit the subscribe button and share this podcast with your friends and family. I'm your host, Catherine Cummings, and you just listened to Healthcare Voices.